Thank you very much to my nominators and to the selection committee. It is truly a great honor to be here and quite intimidating, I must say. So, it's a great opportunity also to be able to give you a brief overview of what we are doing in the lab regarding trying to identify and also overcome innate immune barriers that we think are hampering therapeutically efficient hematopoietic stem cell gene engineering. So before going on, these are my disclosures. So what my lab is interested in particular at is looking at what we call cell intrinsic innate immunity in the context of hematopoietic stem and progenitor cells. So these are antiviral responses that most mammalian cells possess to try and defend themselves against infections. And they are mainly based on the recognition of nucleic acids that we call here triggers that will be sensed by specific molecules in the cytoplasm or in the nucleus of the cells. And then this will activate an antiviral program that is mediated by effector proteins. So this is kind of a black box in the context of hematopoietic stem cells because not much of this has been looked into in the past years. But we really think that this is something important for the field of gene therapy because if you think of it, most of the tools that we use to genetically manipulate these cells are actually going to bound to expose these cells to nucleic acids and most often also to viral vectors. So these are all danger signals that can be potentially sensed by this intrinsic immunity and potentially trigger harmful responses. So we really think that by better understanding these vector-host interactions and innate sensing in hematopoietic stem and progenitor cells, we can actually find means to maximize the efficiency of our genetic manipulations while preserving the biology of these cells. So one of the first things that we started to look at in the lab is to try and understand how hematopoietic stem and progenitor cells actually react to different kinds of gene therapy vectors. So what we've learned from these studies basically is that if you use different kinds of vectors, you will get different kinds of responses from these cells. So essentially, lentiviral vectors, whether they integrate or not, as well as AAV, are going to trigger DNA damage responses in these cells. And this is based on the nuclear recognition of the vector DNA that will trigger p53-mediated transcriptional responses. And we can measure this by tracking, for example, p21 expression levels within our cells. And you can see here that AAV6 as well as lenti quite robustly trigger these kind of responses. And this is exquisitely dose dependent. So the more vector you put on your cells, the more activation of these kind of pathways you will achieve. And this has also nicely been demonstrated recently by my colleagues at SRTjet in the context of gene editing. So you can see here that the amount of p21 expression is very much related to the presence of the AAV6 donor template in this setting. So there are also vectors that will trigger type 1 interferon responses in hematopoietic stem and progenitor cells. And thus far we have identified these vectors to be gamma retroviral vectors, so the first generation gene therapy vectors. And it's interesting to notice that while lentiviral vectors are very efficient in avoiding this kind of activation in human hematopoietic stem cells, gamma retroviral vectors really robustly activate these pathways. And we are currently trying to understand better how this happens from a molecular point of view, what is making the difference between lenti and gamma retroviral vectors. But what I want to mention to you today is that this is something very species specific. So the lentiviral vector avoids very robustly these responses and efficiently these responses in human cells. But if you use lentiviral vectors in the context of murine hematopoietic stem cells, you will actually do much worse using a lentiviral vector compared to a gamma retroviral vector regarding activation of type 1 interferon responses. So this is maybe something that one should consider, for example, working on murine models of disease or just working with murine cells, because some of this signaling could actually give you a confounding readout in your experimental settings. So this signaling also has functional consequences to the cells. So for now we have been focusing on what happens upon the activation of the p53-mediated responses. And in particular, what we have shown is that transduced cells, whether they are exposed to lenti or AAV6, experience more ex vivo apoptosis during the manipulation of the cells. And importantly, the cells exposed to vector are also able to engraft to a lesser extent compared to the untransduced controls. And this is particularly true at early times after transplantation. So if you look at longer time points, there are no longer any differences between untransduced and transduced cells, and this is good news. But obviously, this might have an impact on the kinetics of re-engraftment of the cells within patients. So the good news is that we can actually do something about this by transiently preventing the vector signaling. And as depicted there, here, no pointer, in the image, we can actually target, for example, the apical ATM kinase that is responsible for the p53 phosphorylation in this setting. And we've shown that by transiently inhibiting the ATM kinase during transduction process, we rescue both the ex vivo apoptosis as well as, importantly, in the engraftment of the short-term engrafting cells. So this is something that could potentially be considered as an advantage in the context of transplantations, as it might lead to a faster recovery from the dangerous window of neutropenia that these patients experience. So all of these studies for now have been done in the context of healthy cells, but we are obviously interested also in understanding what would happen in the context of diseased hematopoietic stem cells, because we think that the pro-inflammatory background of some of the diseases or intrinsic genetic defects that the cells have could actually lead to different kinds of responses in these cells. And in particular, we have been focusing on trying to understand vector signaling in the context of Fanconi anemia as well as CGD. So I would just like to show here some of the preliminary results that we have obtained recently in the lab, looking at transcriptional responses of mobilized peripheral blood-derived cells from CGD and healthy controls that have been exposed to a lentiviral vector that expresses the GP91 FOX gene that is defective in these patient cells. And from this transcriptional analysis, we noticed, looking at the differences in response of the cells between healthy cells and diseased cells, is that indeed in this context, there is also a lot of type 1 interferon activation going on in the CGD patient cells. And this was also confirmed by qPCR here below, where you can see that in healthy cells, lentiviral vectors, as shown also before, do not activate type 1 interferon responses, but will do so actually quite robustly in the context of CGD cells. And this is despite the fact that we actually obtained quite low copies in this experiment. So this is something that we really think underlines the importance of trying to firstly improve strategies to prevent vector signaling, but also to think of tailoring them to specific disease context. And we are working on this both in the context of gene addition, but also in editing and disruption approaches. So I would like to now switch gears and go to the part of the effector molecules that are acting in this intrinsic immunity in response to viral infections. And in the field of virology, we call these factors restriction factors. And our idea is that these restriction factors could actually play some role in the fact that hematopoietic stem and progenitor cells are actually quite difficult to modify genetically. And this indeed results in the fact that we still need to pre-stimulate these cells ex vivo, prolonging the time in culture, and also use quite high vector doses, leading to potential toxicity as well as has very obvious implications in terms of cumbersome and costly vector production. So our approach in the lab to try and identify potential restriction factors in hematopoietic stem and progenitor cells has been to kind of work backwards from trying to identify first protocols of transduction that actually work better compared to the standard one. And we and others have identified quite a number of transduction enhancers that efficiently increase lentiviral vector transduction in hematopoietic stem cells. But in particular, we got interested in our observation that cyclosporin A enhances lentiviral vector transduction in hematopoietic stem cells. This is because this was kind of a weird observation. So because in the field of HIV-1, this molecule is quite well known to actually inhibit infection. And indeed, this is something that we see too in the context of differentiated hematopoietic cells, such as T cells or monocyte-derived macrophages. So here you can see that if we transduce T cells in presence of cyclosporin A, we actually see lower efficiency of transduction. And the reason why this happens is actually quite well described in literature. So what cyclosporin A does is to disrupt a beneficial interaction between the incoming vector capsid and this host factor called cyclophilin A. And this is going to lead to premature uncoating of the vector and thereby inhibit infection or transduction. So we got really interested in trying to understand how come in hematopoietic stem and progenitor cells we actually see exactly the opposite. So in trying to figure out what cyclosporin A actually does in stem cells, we realized doing experiments that it's potentially doing a number of different things. So one of our first observations was that if we use an integrase defective lentiviral vector, cyclosporin A doesn't give any advantage to transduction. So this was kind of suggesting to us that maybe it's acting at the level of the vector integration. However, then we noticed that if we start to fiddle with the vector capsid, so we can use different kinds of capsid mutants to build our vectors, and here we have used a capsid that no longer requires the host factor cyclophilin A. Well, in this context, cyclosporin A starts to give an advantage also to non-integrating vectors. So this was kind of highlighting that potentially this molecule is doing something also early on during the vector life cycle, but that it's simply masked by this negative impact that it has on this beneficial capsid CPA axis with a normal vector. Another thing that we learned from these experiments is that the immunosuppressive arm of cyclosporin A is unlikely involved, because if we use FK506, which is a molecule that inhibits the same calcineurin pathway as cyclosporin A does for its immunosuppressive activities, well, we no longer have enhancement of transduction. So based on these results, we kind of reasoned that if we would be able to identify a cyclosporin that is not immunosuppressive, so would potentially be less toxic, and would not be binding this host factor cyclophilin A, so we could get rid of this first negative impact that it has on the vector, then we might actually be able to reach even higher efficacy. So indeed, Carolina in the lab searched the literature for these kind of derivatives of cyclosporin, and she identified a cyclosporin that really nicely meets these requirements, and it's called cyclosporin H. So she tested cyclosporin H in our stem cell transduction protocols, and indeed, as predicted by our observations, cyclosporin H is an even more potent enhancer of transduction compared to cyclosporin A. And this was actually quite remarkable. So this is an experiment in which we actually titer the vector in CD34 positive stem cells. So we usually do this in 293Ts, and this gives us a reference titer that you can see here in the white bar. Then if we titer the same vector in CD34 positive cells without any enhancement of transduction, we usually get around log-less vector because of this intrinsic resistance these cells have to transduction. So this is the gray bar. However, if we titer the vector in stem cells in presence of cyclosporin H, which is the orange bar here, it really nicely rescues the vector titer to the levels that we get in the very permissive 293T cells. So this was quite remarkable, and we hadn't really seen anything like this before in our hands. So of course it was very important for us also to show that this kind of enhancement is maintained in long-term repopulating cells in vivo. So I'm just going to show you some highlights of our experiments in mice. And what we've done here is to use mobilized peripheral blood-derived cells and a clinical grade vector, and we have compared the one-hit transduction protocols in presence or absence of cyclosporin H to a two-hit standard transduction protocol. And you can see here on the left side, these are the copies that we retrieved from the marrow of these mice long-term, and cyclosporin H mediated enhancement is quite remarkable. So we obtained around log higher copies compared to the one-hit control in vivo long-term, and this amounts up to a twofold increase compared to the two-hit standard control. And this happens without affecting the engraftment capacity of these cells, which is of course very important. So now that we had a molecule at hand that wouldn't be acting negatively on the first phases of the vector entry into the cells, we reasoned that this cyclosporin could actually give an advantage also to a normal IDLV. And indeed, this is the case. So we reasoned that this could be useful to test in the context of gene editing approaches, where you can sometimes choose to use an IDLV as a donor DNA template. So we tested this idea, and indeed this is just a highlight of the results. These are vector copy numbers, editing efficiencies that we retrieve from mice long-term that receive the edited cells. So there is no engraftment impact on engraftment of these cells, but you can see here an up to 4-fold increase in the editing efficiencies long-term using Cyclosporin H and the IDLV donor. So as many of you probably are working with an AAV donor in this context, I must say that we noticed that Cyclosporin H doesn't work for AAV6. So potentially this compound wouldn't be giving you an advantage. So I mentioned to you before that Cyclosporin H doesn't impact the long-term engraftment capacity of hematopoietic stem and progenitor cells. However, what we noticed something when we were looking at the peripheral blood engraftment in our mice, and as expected, the one-hit carrier control gives an advantage to engraftment because the protocol is shorter, so the cells stay ex vivo for a shorter time, and so we preserve the fitness and engraftment capacity of these cells. But we noticed that for the cells that had been exposed to Cyclosporin H, we kind of lost this benefit. So the cells engrafted to similar levels as the two-hit standard control. So this kind of reminded us of what we'd seen before in the context of vector signaling. So as I showed you before, we know now that transduced cells actually tend to engraft less into mice, in particular at shorter times after transplantation. So we wondered whether the fact that with Cyclosporin H we enhance so much the amount of vector that actually gets into these cells that we are just increasing the vector signaling so that it kind of compensates for this engraftment benefit. So we tried to repeat an experiment in vivo using lower vector doses. So in this context we used a suboptimal or limiting vector dose of a multiplicity of infection of 20. And as you can see, indeed, in terms of engraftment in the peripheral blood, there are no longer differences between the two conditions. And again, in the bone marrow, the engraftment levels between the two conditions are very similar, but I really want to highlight here the dramatic advantage that we have in this context with limiting vector doses using Cyclosporin H in vivo engrafting cells. So we reach around 80-90% of transduction efficiency with vector copies around 2 copies per cell, while the DMSO control has a really hard time getting any vector into these cells. And this is also true for secondary transplantation, so we took these cells and put them into secondary animals, and the engraftment levels are very similar between the two groups, and we maintain this advantage in transduction efficiencies also in long-term repopulating cells in these animals. So we noticed another thing regarding engraftment of stem cells using Cyclosporin H, and that is that if we completely remove the vector from the picture, so these are mobilized peripheral blood-derived cells that have not been transduced with anything, just exposed to our transduction enhancer Cyclosporin H. And this is a preliminary experiment. It wasn't really done for this purpose, but there is a trend of these cells to actually engraft even to higher degree compared to the carrier control. And this was kind of an interesting observation, also because we have observed in the context of Cyclosporin A, so our old friend, that we tend to see this kind of benefit in engraftment also in the context of transduction. So these are bone marrow-derived cells that have been transduced with a clinical grade lentiviral vector at an MOI of 100, and we see a benefit both in peripheral blood, but as here depicted in the bone marrow of the mice at long term. So the cells that have seen Cyclosporin HA in this context, in red, tend to engraft to a higher degree compared to the two-hit control. So we were wondering whether or not the cyclosporins could actually be doing something more than just enhance transduction in stem cells. And one of the things that we've taken a look at so far is to see the impact of Cyclosporin H on the global proteome of hematopoietic stem and progenitor cells. So we've done this experiment in 3 different cord blood-derived CD34 positive donors in which we can identify proteins that are significantly modulated by the compound. And if we look at the group of proteins that are upregulated by Cyclosporin H, interestingly, we actually pick up a lot of metabolic pathways. So we think this is something that will be worthwhile investigating in the future to see whether or not these compounds could actually have also some biological impact, beneficial biological impact on the stemness of these cells. So what we've learned from these experiments so far is that basically now that we are getting better at enhancing transduction, we might actually start to need getting more worried about the vector signaling. So indeed, we expect with shorter ex vivo culture protocols to obtain a gain also in terms of engraftment, but this can be potentially counterbalanced by increased vector signaling, as I've shown you in the context of cyclosporin H. So one idea could be that we could actually try and combine the two approaches of enhancing transduction as well as transiently getting rid of this vector signaling in order to try and maximize the benefit both in terms of gene marking as well as engraftment of the cells. And also, based on our CGD observation, for example, we might really need to think of tailoring these approaches for specific diseases. So I just want to show you here a very preliminary experiment that we've done in the lab to try and see as a proof of concept if we can actually do this. So we have combined in this experiment our cyclosporin H enhancer of transduction together with this ATM inhibitor that transiently prevents the vector signaling. So this experiment was done with a limiting multiplicity of infection of 20, so we don't really see big differences in terms of engraftment of the cells on your right here. But if you look at the vector copies on your left, we actually noticed that the combination of the two approaches seems to give even higher transduction efficiencies in vivo engrafting cells. So this might seem a bit provocatory, but this is just to show that this is something that we could think of doing, and maybe in specific context or with very difficult to use vectors, this is something that should be considered. So in the last part of my talk, I would like to focus more on what we have learned about the molecular mechanism of action of cyclosporin H, and this brings me back to these effector molecules that I mentioned to you at the beginning. So in order to try and address how cyclosporin H actually enhances transduction in stem cells, we took advantage of something that was around in literature that showed that if you pre-expose THP1 cells to type 1 interferon, we can actually kind of mimic this capacity of cyclosporin A in this case to increase HIV infection. So we wondered whether the type 1 interferon pathway could have something to do with this. And one of the first things that we did was to confirm that cyclosporin H indeed is able to rescue this type 1 interferon-mediated restriction in THP1 cells. So you can see here that in presence of interferon, there is a reduction in transduction efficiencies that is fully rescued in presence of cyclosporin H. One thing that we noticed during these experiments is that this is dependent on the type of envelope that you use to pseudotype your lenti. So it works with a VSV-g pseudotype, but if we change the pseudotype of the vector and use, for example, an amphitropic envelope or this BIFTR envelope deriving from an endogenous retrovirus, and both of these envelopes actually fuse at the plasma membrane, well, first of all, we lose the antiviral effect of type 1 interferon, and importantly, we lose also the capacity of cyclosporin H to enhance transduction. So we confirmed this also in hematopoietic stem and progenitor cells. So here you can see that type 1 interferon decreases transduction efficiency in these cells, so they are responsive to this antiviral cytokine, and cyclosporin H is able to rescue this. And also in this case, this is again envelope dependent, because if we use the BIFTR pseudotype lenti, we lose both the antiviral effect of type 1 interferon, but also the capacity of cyclosporin H to enhance transduction. Of course, the big difference here between hematopoietic stem cells and THP1s is the fact that in hematopoietic stem cells, cyclosporin H is able to enhance transduction also at steady state. So it is the third graph to your right, the white bars, compared to THP1 cells in which you need to pre-stimulate them with an antiviral cytokine to get this effect. So this was kind of suggesting to us that maybe in stem cells, we actually have already expression of an antiviral factor that can be induced in other cell types. And this really nicely fits with a concept that was recently published by the lab of Charles Rice that says that somatic stem cells, not only hematopoietic stem cells, tend to express higher levels of these antiviral interferon-stimulated genes. So basically, we were looking for a type 1 interferon-inducible factor that would likely be acting at the level of vector entry and would be inhibiting VSV-g-mediated entry, but not efficacious against, for example, amphitropic envelopes. So looking in literature for factors that meet these requirements, we were caught by this family of antiviral factors called interferon-induced transmembrane proteins, and in particular, IFITM3 really nicely fits these criteria. It's a broadly acting antiviral protein that acts at the level of the virus entry. It works against VSV, but it doesn't inhibit, for example, amphitropic retroviruses. So we tested the role of IFITM3 in our observed effects of cyclosporin H, and I'm just going to cut to the chase here and show you the data that we obtained in hematopoietic stem and progenitor cells. So basically, in hematopoietic stem cells, what we show is that if we overexpress IFITM3, we get a reduction in transduction efficiencies. Conversely, if we knock it down, we get an increase in transduction efficiencies. And this actually really nicely parallels with the effect of cyclosporin H. So the more the cells express IFITM3, so the overexpressing ones, the more they will benefit in terms of fold increase from cyclosporin H. Conversely, if we knock it down from the cells, so the last column, we almost completely abrogate the capacity of cyclosporin H to enhance transduction. So we had the opportunity to test this also in a cleaner setting using murine hematopoietic stem and progenitor cells that we isolated from the marrow of IFITM3 knockout mice. And you can see that, looking at the white bars, that these cells are actually more permissive to transduction compared to the wild-type counterparts. And importantly, we lose the capacity of cyclosporin H to enhance transduction in this setting in absence of IFITM3. So it really looked like IFITM3 would be the restriction factor or one of the restriction factors counteracted by cyclosporin H in hematopoietic stem and progenitor cells. So how does this work? We don't really have an answer to this question, but what we know so far is that somehow cyclosporin H leads to the transient degradation of IFITM3 in these cells. We can quantify this in terms of immunofluorescence imaging as well as by Western blot. You can see here we have a decrease in IFITM3 levels upon cyclosporin exposure, both at steady state as well as upon interferon alpha stimulation in hematopoietic stem and progenitor cells. And I have to say that this is a protein effect. So we don't see any impact on the messenger RNA levels of IFITM3 in presence of cyclosporin H. And as I mentioned to you, this is also a transient effect. So if you wash away the drug from the cells, you will have a very quick rebound of the expression of this antiviral factor in your cells, as well as the negative effect it will have on the transduction efficiencies in your cells. So this is something to keep in mind in case you are interested in trying this molecule in your protocols. You really need to keep cyclosporin H together with the lentiviral vector throughout the transduction period to get maximal efficacy. So another thing that we've learned, and this is still ongoing, is that there are probably cell type specific factors that are required both for the antiviral activity of IFITM3 and the capacity of cyclosporin H to degrade it. And this is because we actually can recreate really nicely all of these phenotypes in THP1 cells. So you can see here that overexpression of IFITM3 is going to restrict transduction efficiency, and this is fully rescued by cyclosporin H in the graph on your left. And in the Western blot, you can see that cyclosporin H really nicely degrades IFITM3, both at steady state and upon interferon alpha stimulation in THP1 cells. However, if we change cell type and try to recapitulate this, for example, in K562 cells, nothing really happens. So if we overexpress IFITM3, it's not restricting transduction, and cyclosporin H has no effect. And interestingly, we also noticed that cyclosporin H is not able to degrade this factor anymore in these cells. And this is not because K562 cells do not respond to type 1 interferon. They do, and you can actually see also from the Western blot that we really have a nice upregulation of IFITM3 levels in presence of interferon alpha. So there is something more to this. And this dichotomy between the phenotypes that we get in THP1s, K562, and also in stem cells has allowed us to set up a number of approaches to try and identify these cofactors. And we are currently trying to identify both IFITM3 as well as cyclosporin H interactors, specifically in hematopoietic stem cells, taking advantage also of these cell line models that we have. So finally, I would like to show you these last results that we've obtained looking at IFITM3 in hematopoietic stem and progenitor cells. And an observation that we made was that individual donors of hematopoietic stem cells actually express quite different levels of this antiviral factor. So this is the upper panel. And this negatively correlates with transduction efficiencies. So the more IFITM3 your cells will have, the less transducible they will be. The good news is that Cyclosporin H benefit in terms of fold increase of transduction efficiencies actually correlates with this in an opposite manner. So the more restricted and difficult to transduce your cells are, the more they will benefit from cyclosporin H in terms of fold effects on transduction. So this kind of suggests that cyclosporin H could be a kind of a special transduction enhancer because not only would it be enhancing transduction, but it also could have the potential to abrogate donor variability. And also based on our preliminary results regarding the effects on engraftment, it could potentially also preserve hematopoietic stem and progenitor cells during the ex vivo transduction protocol. And we are, of course, also interested in looking at more into the potential role of IFITM3 in donor variability, which remains a really critical issue in gene therapy. And we are interested, firstly, in trying to identify what actually regulates IFITM3 levels in these cells. And SNPs are one of our candidate hypotheses in this context, and we will have the unique opportunity to test this in the context of gene therapy patients within our institute, but we would also be very interested in trying to collaborate with with other research centers. So I hope to have given you some hints and results on how we really think that understanding innate immunity and the vector-host interactions in the context of hematopoietic stem and progenitor cells will allow us to design improved strategies, molecules or approaches that will help us gain efficiency of all of our gene manipulation platforms in terms of efficacy, but also in terms of preserving the biology of the cells, with the idea that we can actually find a sweet spot between increasing efficacy, maintaining low the vector signaling, and not activating innate immune responses. So of course this will have hopefully direct implications in terms of translational research, but also more from a basic biology point of view, gaining information on host virus interactions as well as stem cell biology. So finally, this is my last and most important slide. I really need to acknowledge many people from SRTJET, in particular Luigi Naldini, who recruited me back in 2012, giving me the opportunity to start working on innate immunity and virology in the context of gene therapy. Many external collaborators, but in particular I need to thank all the wonderful young scientists, very dedicated people that work with me in the lab every day, and in particular Carolina and Julia have been the ones that have been driving this IFITM3 cyclosporin H part. So Carolina is now a postdoc. She moved to New York at Columbia University, still working on stem cells in Hans Snöck's lab. And Francesco Piras, together with Erika Valeri, have been the ones driving more the vector signaling and innate sensing part in stem cells. So we are always looking for motivated postdocs, so if you're interested in innate immunity and virology in the context of HSPC gene therapy, catch up with me. I need to thank my funding agencies and of course you for your attention. Thank you.
Thank you, Dr. Rudnitski. So now the floor open for question for Anna, please. Yeah, one question.
Great talk, thank you. So I guess nobody is not sure about what triggers this interferon signaling in the system, but I think there's a gap in the system because when you think about this system that is consists of one single cell type, then we have to have this interferon signaling, especially you need actual producer of interferon molecule itself. So how do you correct that gap in your system?
So we are not actually measuring interferon in these cells, but hematopoietic stem and progenitor cells can actually produce interferon upon infection. These are intrinsic immune mechanisms that are independent of the cell type. So most mammalian cells are capable of these kind of responses, and it turns out that also stem cells do this. And so what we see is that especially with gamma retroviral vectors, we have the expression, so what we measure is type 1 interferon stimulated genes that are kind of a marker of this response. But we have shown for gamma retroviral vectors that if we block the interferon alpha receptor, we can abrogate this response. So it suggests that we need to have a paracrine effect of this cytokine. And what we know also, at least in part, in particular for the p53 responses, is that they actually occur in all the different stem cell subsets. So this is not more about the progenitors or the more primitive cells. All of these cells are able to mount these kind of responses. I don't know if I answered your question.
Yeah, that was really good answer. Related question, would you consider RIGI or MDA5 or STING as the upstream of your IFTM 15 or 30?
Yeah, that's a good question. We are investigating that. We have some indications, actually, that it is not, we initially thought that it's the RNA of the gamma retroviral vector that triggers the type 1 interferon responses, because if we block reverse transcription, so we don't have the DNA of the vector, we have the RNA genome, we still get these responses. But we have now new data showing that it's likely more not the genome but structural components of the vector that are triggering this response. So we don't know what the sensor is currently.
Thanks.
Anna, can you comment on when you increase transduction efficiency of integration deficient lentis? Is
Sorry.
that when you increase the transduction efficiency of integration deficient lentiviral vectors by using cyclosporin H, do you increase the expression level in a given cell, or do you increase the percentage of transduced cells, or both?
I would say both. So tendentially, we increase the percentage of transduced cells, and we've done these kind of studies actually more in the context of cyclosporin A, that show that cyclosporin A, differently from other enhancers such as rapamycin, tends to increase more evenly the percentage of transduced cells rather than increasing a lot of copies in a lower fraction of cells. So I could infer potentially that cyclosporin H does this kind of thing too, but we haven't really looked into that carefully, but my guess would be that until you reach saturation, it's going to increase more the percentage, and then probably you will start building copies.
Okay, I was asking because there is a lot of reported variability when you compare integration proficient and integration deficient vectors. The cell type that is transduced makes a very big difference, and I just wonder whether that variability was partly related to this. Just a comment rather than a question.
Yeah, so we don't actually, so you are asking why cyclosporin A doesn't work for ideal V? No.
No, I was wondering whether this effect that you are describing may partly explain why different cell types express equally from proficient and deficient vectors or not.
Okay, we haven't investigated this in detail. I don't think we get silent integrations.
Thank you.
Okay, if there's no further question, let's give Anna another round of applause to congratulate her.